Hi everyone, welcome back to Biological Psychology. Today I'm going to cover Chapter 18, Brain Mechanisms of Emotion. Today I'm largely going to talk about the limbic system. Now this is a system of different structures in the brain that are involved in many different neural processes. But the limbic lobe originally got its name from French neurologist Paul Broca. Now, according to Paul's definition, he believed that the limbic lobe consisted of the cortex, mainly the cingulate cortex shown in blue here, outside of the corpus callosum, and then the cortex on the medial surface of the temporal lobe, in addition to the hippocampus. And although Paul Broca didn't originally think that this area was important for emotional processing, he primarily thought they were involved in olfaction. It wasn't until the 1950s where scientists started looking at how this limbic system was also utilized in emotional processing. So, what brain structures are involved in emotion? Well, in the 1930s, Dr. James Pepys suggested that several structures in the limbic system, which again was first introduced by Paul Broca, played a role in emotional processing. He believed that our emotion system was linked between the cortex and the hypothalamus. And of course, in neuroscience and psychology, some of the ways that we can discover the purpose of a brain area or the function of a brain area is through injury. And following damage to certain cortical areas, like the cingulate cortex, doctors and physicians have found this changes emotional expression in their patients with little change in perception or intelligence. And so Dr. Pepys believed that the cortex was highly involved in experiencing emotion and processing emotional memories. And this system was named the Pepys circuit after Dr. James Pepys. Now, as I previously noted, Dr. Pepys recognized that patients that have damage to some cortical areas may have different emotional experiences and may have changes in their emotional expression. He also studied the effects of tumors in the cingulate cortex. And tumors in the cingulate cortex are sometimes associated with changes in emotion or emotional disturbances like fear, irritability, and depression. So ultimately, Dr. Pepys proposed that the cingulate cortex is playing an important role in processing emotional experiences. And ultimately, information in the cingulate cortex would be sent to the hippocampus. So he proposed the hippocampus was also a part of this circuit. We can think of the hippocampus like a loading dock for information. The hippocampus is going to take newly formed memories and help us consolidate it to potentially become a long-term memory. The fornix is this important area that connects the hippocampus and the hypothalamus. And neurons that are going to be synapsing onto the hypothalamus, Dr. Pepys believed played a role maybe in emotional expression. Ultimately, the hypothalamus communicates with the anterior nuclei of the thalamus, which will then go back to the cingulate cortex. So ultimately, Dr. Pepys proposed this circuit and the activation of these key brain areas, many of which are a part of the limbic system. And he proposed that these key brain areas played an important role in emotional processing and the consolidation of emotional memories. Now, because of the similarity between the limbic system and the Pepys circuit, the structures in the Pepys circuit are often referred to as the limbic system. As I mentioned previously, if you had a tumor in the cingulate cortex right here in blue, this has been associated with certain emotional disturbances like fear, irritability, and depression. So ultimately, Dr. Pepys suggested that the hypothalamus governs the behavioral expression of emotion. And he proposed in his circuit that the cingulate cortex affects the hypothalamus via the hippocampus and the fornix. And don't forget the fornix is a large bundle of axons that leave the hippocampus to allow it to communicate with other structures outside of the hippocampus. Ultimately, he proposed that there's this bidirectional relationship between the cingulate cortex and the hypothalamus due to the anterior nuclei of the thalamus. Now, we know the thalamus is the brain's key relay station, and there are important structures we've talked about, like the internal capsule, that connect the thalamus and the cortex. So ultimately, this is the Papes circuit that Dr. Papes introduced. So now we're going to talk about the brain and personality. So as I mentioned previously, sometimes accidents or traumatic brain injuries can help neuroscientists and scientists discover the importance and function of certain areas of the brain. And that is the case with the story of Phineas Gage. So his story really tells us that brain damage can profoundly influence our personality. So Phineas Gage was around in 1848, and he was working on a railroad in Vermont. There was an explosion at the railroad, and unfortunately, this rod that you see here, it's about 1 meter long, actually went through his head. So this rod went through his head just below his left eye right here, and it passed through his frontal lobe right out of the top of his head. And the hole through his head was more than 9 centimeters in diameter. So miraculously, Phineas Gage did end up recovering from the accident. It's amazing that he didn't have some terrible infection after having this gaping hole in his head, but he did go back to normal, and everything was normal except for one thing. His personality drastically and permanently changed. Now, remember where the iron rod went through. It went through his eye and into his frontal lobe. Today, we know that the frontal lobe plays a really crucial role in our personality, our decision-making, and our planning. After the accident, his personality was very different. His friends and family reported that he indulged in profanity, that he was very impatient of restraint or advice, and at times he was very grumpy and more aggressive than he normally had been. When he tried to return to his old job as a construction foreman at the railroad, they actually denied him. They wouldn't rehire him because his personality had changed, and it was difficult for him sometimes to plan out tasks and to complete tasks. Interestingly, I don't know why he'd want to go back to the railroad after having such a traumatic experience, but that's just my thought. Phineas lived for another 12 years, but when he died, there was no autopsy performed, unfortunately. But if you're interested in his story, you can actually visit his skull and the iron rod that went through his head. It's at Harvard's Medical School Museum. It's interesting to note that the iron rod severely damaged the cerebral cortex in both hemispheres, particularly in the frontal lobe. And it was due to this damage in this key brain area that potentially led to Gage's emotional outbursts and these drastic changes in his personality. As mentioned previously, the amygdala plays a crucial role in processing emotion. Now, due to bilateral symmetry of the brain, if we were to cut down the longitudinal fissure and have 2 sagittal sections of the brain, we would see the amygdala on both sides. And the amygdala sits in the more anterior area to the hippocampus. Ultimately, the amygdala has been known to play a special role in fear at memory processing. When we explore the evidence connecting the amygdala to fear, I want you to keep in mind that there are other brain structures that are also activated when we are in fear. But the amygdala is also active in other emotional states. So it's not just playing a role in fear processing, it's playing a role in other emotions. So interestingly, after the Papes circuit was further explored by Dr. Papes, and he proposed that this was an important emotion circuit in the brain, other neuroscientists wanted to further explore how the brain processes emotions. And so Kluver and Bucy were two neuroscientists following James Papes who did this. So they conducted a series of experiments in rhesus monkeys, and they actually conducted a bilateral removal of the temporal lobes, meaning they removed the temporal lobes from both hemispheres of the brain. And interestingly, they found that this had a dramatic effect on the animal's aggression and fear response. This procedure led to many behavioral abnormalities that I'm going to talk more about on the next slide, but this is referred to as Kluver-Bucy syndrome. Now, this is very rare in humans, but Kluver-Bucy syndrome can present symptoms in certain cases in humans. So, for example, herpes simplex encephalitis has been shown to primarily affect the temporal lobe. And more commonly, trauma, stroke, or Alzheimer's disease can also affect neural processes in the temporal lobe. For example, in Alzheimer's, we know that there is a severe loss of neurons in the hippocampus, and the hippocampus is in the medial temporal lobe. So ultimately, that shrinkage of the brain that's known as brain atrophy could induce these symptoms of Kluver-Bucy syndrome in a human. And this is ultimately because we know the amygdala helps us regulate emotion, and it reinforces behaviors related to food and sex. So in a moment, I'm going to give you some examples of how total loss of the amygdala and a bilateral removal of the temporal lobes affected behavior in the rhesus monkeys. Following the removal of the temporal lobes, the rhesus monkeys in Kluver and Bucy's experiments basically exhibited decreases in fear response and decreased aggressive behavior. So ultimately, there was a corresponding decrease in their vocalizations and facial expressions that were usually associated with fear. So for example, monkeys are usually innately afraid of snakes. And so even when presented with something that would usually induce fear in the monkeys, due to this bilateral removal of the temporal lobe, they no longer responded to snakes and other objects like this with the normally associated fear. And virtually all of these symptoms in rhesus monkeys have been seen in humans that have lesions in the temporal lobe, or specifically lesions in the amygdala. So ultimately, people who have lesions in the amygdala appear to have this flattened emotional response. So let's talk more about the amygdala. So it's situated in the pole of the temporal lobe just below the cortex on the medial side. And as I mentioned previously, due to bilateral symmetry, we can see the amygdala in both hemispheres of the brain. It sits in this anterior view to the hippocampus, as shown here. So ultimately, the human amygdala is made up of three different groups of cells, including the basolateral nuclei, the corticomedial nuclei, and the central nucleus. The amygdala receives afferent signals from a large variety of sources, such as the neocortex, as shown right here. And remember, the neocortex is a part of the Papes circuit, as well as the hippocampal and cingulate gyri. And ultimately, information from all of these sensory systems feeds into the amygdala, particularly here in the basolateral nuclei. And the amygdala also communicates with the hypothalamus as they are in close proximity to one another and rely on that communication for emotional processing. So now let's talk more about amygdala activation. I'm going to talk about a series of experiments. This was really interesting. It was conducted by Hamann and colleagues in 1999, and essentially they conducted PET imaging to look at brain activity. If you remember, this is a commonly used tool to look at metabolism and activity in the brain. And so human participants in the study were randomly selected to look at a series of pictures. So they were either asked to look at pleasant images, so this could be something that was appealing or arousing, perhaps an appetizing food. Some were asked to look at a frightening or aversive image, such as a scary animal or a violent scene. And then others were just asked to look at neutral pictures. This served as a control group. So they were just looking at a house or plants. Interestingly, the results of the study demonstrated that compared to the neutral subjects, human participants who saw pleasant or unpleasant stimuli evoked greater activity in the amygdala. And as you can see here in this imaging, the amygdala was activated from this emotional stimuli that was either pleasant or unpleasant. And in the second phase of the experiment, the subjects were brought back and put into the PET machine and shown a variety of pictures. And they were asked to use their memory and identify which pictures they had seen in their initial conditioning session. And ultimately, those who were able to recall the images were the ones that looked at the emotional pictures rather than the neutral ones. And this makes sense because we have enhanced memories for emotional pictures. So this correlated with increased amygdala activity. We know that brain lesions can alter emotional behavior, and clinicians have tried to conduct surgeries to treat behavioral disorders in humans. And one that's pretty well known, but it no longer is utilized, is called the frontal lobotomy. Today, it's really difficult to imagine that someone would conduct a lobotomy because you're essentially destroying a large part of the frontal lobe of the brain. But at one point, this was thought to be therapeutic. So in 1949, Dr. Moniz actually won a Nobel Prize for his technique called the frontal lobotomy. And although there are no lobotomies that are performed anymore, tens of thousands of lobotomies were actually performed during World War II. So usually, this would be a procedure to try to help reduce severe behavioral disorders or psychiatric symptoms. So in this procedure, essentially a physician would perform the lobotomy by inserting this tool into the eye, and they would scrape in this brain area, which would sometimes essentially detach the frontal lobe from the rest of the brain. So ultimately, the frontal lobotomy can actually be performed with little decrease in IQ levels or memory loss, but it does induce other strange effects. So these effects are primarily related to the limbic system because frontal lobotomies have shown to blunt emotional responses and induce loss of emotional components of thoughts. Patients who receive lobotomies also developed inappropriate behavior or potentially had lower moral standards. So ultimately, this is kind of similar to Phineas Gage. Patients had considerable difficulty planning and working towards goals. They were often easily distracted and had trouble concentrating. So ultimately, this lobotomy, the frontal lobotomy, is not practiced anymore. Thank goodness we live in the 21st century. But at one time, this was used as a quote, therapeutic technique that physicians thought would help reduce symptoms of psychiatric disorders or traumatic experiences. Now that we've talked about the way that the brain processes fear-based memories and emotions, primarily through the amygdala, but of course there are other structures involved in this fear-based processing, I wanted to briefly talk about aggressive behavior. And so the medial hypothalamus shown here sends axons to another area of the brain called the periaqueductal gray matter, as you can see right here. And the periaqueductal gray matter of the midbrain has been associated with aggressive behavior. Experiments have shown that if you electrically stimulate the PAG, it can actually produce effective aggression, and lesions in this area can disrupt aggressive behavior. Interestingly, the hypothalamus and the periaqueductal gray matter appear to influence behavior partially based on input it receives from the amygdala. So these structures are all connected. And thus, the amygdala may also play a role in the way that aggressive behavior is processed due to its communication with the hypothalamus and the PAG. Aggressive behavior has also been linked to serotonin deficiency. A variety of studies suggest that serotonin plays a really important role in regulating anger and aggression. And this makes sense because we know that serotonin is our mood stabilizer neurotransmitter. So, if you remember from previous chapters, we've talked about how serotonergic neurons, right here, live in the brainstem, and they're referred to as the raphe nuclei. Now, these neurons have long Golgi type 1 axons that project to other areas of the brain to ensure that the brain receives proper serotonin levels. However, there is experimental evidence that supports something known as the serotonin deficiency hypothesis, essentially stating that aggression is inversely related to serotonergic activity. So, for example, if you had reduced levels of basal serotonin, this could potentially increase aggressive or angry behavior. A multitude of studies have found that if you block serotonin synthesis, it can increase aggression in rodents. And interestingly, there are at least 14 different serotonin receptor subtypes in the brain. Several experiments have shown that agonists of serotonin receptor subtypes decrease aggressive behavior. So we know that agonists mimic the naturally occurring neurotransmitter, thus increasing serotonin levels. Where antagonists, so these are drugs that are actually blocking these serotonin receptors in the brain, can actually increase aggressive behavior in mice. And this makes sense because blocking serotonin would reduce the amount of serotonin that's available in the synaptic cleft. This is very similar to what we've talked about with drugs therapies for generalized anxiety disorder and depression. We've talked about SSRIs or monoamine oxidase inhibitors that are therapeutic strategies to increase the amount of serotonin that's left in the synaptic cleft to help boost mood. So it does make sense that blocking these receptors would increase aggressiveness in mice, as serotonin is a mood stabilizing neurotransmitter.